Welcome back to our session. We're still talking calculations involving acid and base reactions in the context of titrations. We managed to see that this actually has a lot to do with stoichiometric calculations, and the calculations involved once a titration has been conducted would describe them as, volumetric analysis. Now, let's look at, some of the important aspects in as far as a titration is concerned. Now, in terms of the terminology, so the technique involved is about the titration. So titration is about the technique, all right, of actually, using a standard solution. This is a technique or a procedure that is carried out using a standard solution to determine an unknown concentration. We spoke of an end point of a titration and a point of equivalence, where we spoke about the chemical indicators which have a color change, okay? So this has to do with the indicators, the end point, okay, where in a neutralization reaction, actually all the substances have actually reacted, right? And then what exactly are some of the apparatus or equipment actually involved in a titration? So what remains important is also for us to be able to identify the correct names of apparatus, or equipment that we use to actually carry out the technique or the procedure in a titration, which involves a reaction between an acid and a base. So identifying the apparatus is another skill that is very, very important, right? What are some of those? We know that there's a barrette, which is actually calibrated to give us the volume of, a solution that was used in a reaction. So this is the barrette. And then we also know of a beaker, okay, which will contain, some substance which has been measured or pipetted. So the pipette actually measures a fixed volume of a solution, all right? And then at the end of the day, when a titration takes place, we know actual volumes of substances or quantities. But remember, one of these solutions in this setup is actually a standard solution. So we'd use that standard solution to actually determine the unknown concentration of the other substance, right? Okay. Now, what exactly is involved in a titration? So before we actually start to titrate or carry out the whole procedure of a titration, we need to select an appropriate indicator, right? So you'd have mentioned, earlier on, the different indicators. So our choice of indicator in an acid-base reaction depends on the type of acid as well as type of what? Base, which is also informed by the pH range in which that specific indicator actually undergoes a color change to show us the end point of this acid-base reaction, right? So we need to write down the colors of the indicators of the indicator in an acid and in a base, and then at the end point. So this is actually what is happening in as far as the choice of the indicator is concerned, right? Okay. Now we also need to measure out the exact volume, right? So I think this goes further to emphasize the measuring skill. For us to keep track of data in a chemical reaction, when we conduct a technique of titration, we need to be able to carry out measurements and record those volumes. Because remember, in the volumetric analysis, that involves the calculation, the maths part of it. We need those actual volumes to actually, so that we are able to quantify or to actually get the unknown concentration of the other substance, either the base or an acid. So in a nutshell, what we are saying is that if the acid is a standard solution, then the technique of titration would involve us determining the unknown concentration of the base, and vice versa. So if the base is also a standard solution, so in that titration, we would actually want to determine the unknown concentration of the acid. Okay, the indicator, very, very important. It shows the color change at the end point of that acid-base reaction. So we also need to add a few drops of the indicator so that we can keep track of the color change in as far as the end point is concerned, right? Okay, so we also have to fill the burette with the solution of the known concentration, right? So what goes into the burette is actually the standard solution, okay? Because this solution we know is concentration. So the burette is actually calibrated, as you can see. It means that there are calibrations. So when it's completely filled, it will mark up to 0 mils, okay? Whatever units are used. So as you allow that standard solution to come out of the burette so that it reacts with the base, it could be the acid or the base, it doesn't matter, and then you can keep measurements or track of how much of the standard solution in terms of volume has been used in the reaction, okay? And also, very, very important, you need to make sure that the bottom of the meniscus, so the meniscus is that curved, you know, part of the liquid surface. So make sure that when you take readings, they're actually correct. And we also need to keep record of volume of solution in the burette, right? And also, we need to place the conical flask under the burette directly so that all the standard solution goes into that, actually the conical flask where there is the other substance. And then we add the solution in the burette and swirl gently. The swirling will actually ensure that at the end of the reaction, all the substances have reacted completely, okay? And then the color of the unknown solution will change in the region where the acid and the base mix, okay? So that's where the reaction actually takes place. And then the color would be at the point. We also take note that at the end point, there is that color change, which will tell us that, okay, this reaction has reached, you know, the end, and then we can take the readings in terms of the volume of the standard solution. So the pipette will be used actually to measure the volume of the solution that goes into the conical flask, right? Okay, so we need to also keep track of the color change. We've mentioned that, right? So as we approach that, you know, endpoint, we need to release drop by drop, you know, just in small amounts, so that we also address the issue of accuracy in terms of the volume of the standard solution. Okay, right? And then also, we need to record the volume of the solution in the burette, that's of the standard solution, okay? And the volume of the, you know, that of the other substance would have been recorded, the one that goes into the conical flask, okay? So it's about, you know, measurement, and more measurement, right? So at the end point, we would actually say, okay, so this is roughly the amount or the volume of the standard solution that was used in the titration, okay? Then would obviously have to repeat the procedure, you know, more than once, say 3 times, you know, because just to get a sense, the first one is basically to give us an, you know, a rough estimate in terms of how much volume of standard solution is actually used. So if we repeat the procedure 3 times and average, that also appeals to, you know, the accuracy of our results. So in any experiment, if we do 3 trials and average them, we get a better sense in terms of the actual or the most accurate readings in as far as, you know, the data that we collected, right? So if you repeat that, that's one way of improving accuracy. So these are some of the most important, you know, aspects in as far as the actual technique or the procedure of a titration. After the short break, we'll try and look at, you know, tangible example, practical examples of a titration, as well as, you know, the calculations in as far as the volumetric analysis. Let's take a short break for now.